the last talk of this seminar 2026 winter edition. Luis could not be here with us today to introduce Luca, so I will take care of introducing Professor Luca Valenti. He's an international recognized expert in the field of hepatology. He is a faculty member in the University of Milan. He also works as a clinician in the Policlinico of Milano. He got his medical studies in the University of Milano, and then he pursued his clinical training to become a specialist in internal medicine and hepatology, and then he completed all this training by doing a postdoctoral stage in Columbia University in New York. At the research level, he's known by contributing in the intersection of metabolic liver disease, genetics, and precision medicine. And he has many achievements in the field of research. He has more than 500 publications in peer-reviewed journals. He has many national, international awards. He's editor-in-chief of Liver International. And what is more important, he's a key opinion leader in the field of genetics in muscle D. Thank you, Luca, for accepting our invitation, and we are looking forward for your presentation.
Thanks so much for the very too kind introduction, and also to you for the invitation, which I appreciate a lot. So the topic of my talk today will be exactly the role of genetics of MESOL D, but with a clinical look. So I'm a clinician, so I will try to show you how the new discovery in the fields of genetics and translational medicine may reshape the clinical management of people with metabolic disorders, and in particular MESOL D in the future. So in particular, I will review how the inherited genetic variation influence the risk of development of the progression of the disease, helping us to move towards an individualized care, focusing in particular on the role of the PNPL3 genetic variant, which is the main genetic determinant for this condition in humans. These are my disclosures. So as you know, we are a bit from also the previous presentation, steatotic liver disease in general has become a global phenomenon. It affects 1 in 3 people in a condition strongly intertwined with the obesity, diabetes, metabolic syndrome or obesity. And the rising prevalence is mirroring that of liver-related complications, including the compensation due to cirrhosis and hepatocellular carcinoma, and it's contributing to the rise of this complication, in particular in Western countries, but even in Asia, the Middle East, and globally. So despite this epidemiological weight, the tools for the early diagnosis, the early recognition of identification of people at high risk of progression towards the liver-related complication of the disease is still limited, still in infancy. We have tools that are able to identify those at risk of liver-related events at late stage of liver disease progression, advanced liver fibrosis, but not since the very early stage, to target preventive, really preventive approaches, not just surveillance for the complication or treatment of the advanced disease. And liver-directed pharmacotherapies are still at the beginning. We have heard before, we have resmetirom and semaglutide, but they are effective in reducing fibrosis, reversing the disease still in a minority, not in all the patients. Okay, so the goal would be to exploit the genetic heritability of this condition to find new biomarkers able to predict the development of liver-related complication, to identify new therapeutic targets to prevent disease progression. So the numbers are in this striking, but beyond the association of MESOL D with metabolic complication disease, the presence of increased hepatic fat content also increases the risk of cardiometabolic complications of the disease and increase the risk of developing diabetes in those who are not yet diabetic. It increases the risk of cardiovascular disease, chronic kidney disease, heart failure, and even is associated with extrahepatic cancer and serious bacterial infections. But what interests us more is the increase in the risk of liver-related complications, in particular cirrhosis and hepatocellular carcinoma, that are those that are relatively more increase in people with mesol D. But we should keep in mind that these are not the leading cause of morbidity and mortality in the population in people with mesol D. It's only a minority of people affected by this condition will develop serious liver-related events during their lifetime. Most of them will have health problems due to cardiovascular disease or extrahepatic cancer. So, the severity of insulin resistance also predict the risk of extrahepatic complication of the disease. So we need more refined tools to identify specifically those that will undergo liver-related complications. We know that the main drivers of this development of progression of liver disease are insulin resistance, other endocrine conditions such as hypothyroidism, hyperglycemia, a sedentary, unhealthy lifestyle. We have heard about sarcopenia, some dietary factor, intake of saturated and trans fats, toxins, fructose, and alcohol. But we also know that genetic variation influences, inherited genetic variation influences the susceptibility to the accumulated steatosis and the progression to the inflammatory form, namely steatohepatitis, that can then lead in about 10% of the patients to progressive fibrosis, advanced fibrosis, and cirrhosis with portal hypertension and liver cancer. And this, of course, interacts with factors related to the microbiota and epigenetic factors that modulate the effects, of course, of genetic predisposition. But we know indeed that mesol D is not solely an environmental disease. In the last years, the family aggregation studies, twin studies, and population large scale analysis all led convergent estimation of heritability at about 50% of the hepatic fat content and steatotic liver disease. So this is one of the most heritable common condition. About 50% of the risk of accumulating fat in the liver and developing a liver disease is not due to metabolic environmental factor, but is influenced by inherited factor. Indeed, not every people with obesity and type 2 diabetes go on to develop liver-related complications, just a minority. And in the last years, the genome-wide association studies led us to the identification of the main genetic determinants of this condition, at least the most important common genetic variants that I've summarized for you here in this table with the 5 most robustly validated variants that are causally associated with the risk of developing progressive steatohepatitis and liver disease. These are all associated except the protective one with an increase in hepatic fat accumulation, which translates into an increased risk of steatohepatitis, including steatohepatitis due to other triggers of fat accumulation, such as in particular alcohol, of cirrhosis, liver cancer, and also translate for those with a larger effect in increased risk of liver-related mortality. So in particular interest are, of course, the variants in PNPLA3, which is the main genetic determinant of this condition due to the moderate to large effect size and to the fact that it's very common in the general population, and variants in TM6SF2, and the protective variant on the other end on this gene called HSD17B13. So in large biobank cohorts, such as in the UK biobanks, these are data from European ancestry participants in the UK biobank cohort. These 5 variants reported in the previous table actually accounts for a large fraction of the variability of the liver disease severe phenotypes, such as cirrhosis in yellow and hepatocellular carcinoma in red in the general population. Here, a population characterized by a high prevalence of obesity and low prevalence of chronic viral hepatitis. Of course, comparable to that of obesity and type 2 diabetes, so these are major drivers of liver disease in the population. But an important role is played, as you can see here, by the PNPLA3 148 methionine variants alone, the variants that I was discussing before, accounting for as much as 18% of cirrhosis variability in the general population and 28% of liver cancer, hepatocellular carcinoma variability in the general population. Meaning that in theory, if we could correct the molecular mechanism alteration caused by this genetic factor in the population, we may be able to, in people with, of course, metabolic triggers of disease, we may be able to prevent a large fraction of the progression to liver-related complications in the general population. And what's more, also this variant has a large variability in the prevalence across different ethnicity globally. The variant allele, the methionine allele associated with an increased risk of steatotic liver disease is here indicated in blue. As you can see here, is highest among Native Americans in South and Central America and in East Asia and in Siberia. It's very high, and this variation in the prevalence of the PNPL3 variant globally accounts for about 50% of the interethnic susceptibility to differences in susceptibility to develop steatotic liver disease, cirrhosis, and liver cancer in the population. So this is a major driver of the risk of progressing towards a severe liver disease globally, not only in Europeans. This is something, unfortunately, for basic and translation studies that is only present in humans. It appeared in hominids about 1 million years ago. And it's now, of course, detected in about 40% of chromosomes in globally, but it was fixed in some other hominids, such as in the Neanderthals and Denisovans, during history, possibly during the glacial era, because the variant which encodes for an amino acidic substitution, we will see, and the PNPL3 protein involved in the regulation of lipid metabolism may favor cold adaptation because it's also expressed and physiologically relevant in the brown and white adipose tissue. So what's important was that in the first genome-wide association studies, the PNPL3 methionine variant was the strongest signal associated with liver enzymes, the hepatic fat content, the risk of histological mesoly, and the progression towards cirrhosis and hepatocellular carcinoma. A key insight of this study was that the metabolic triggers of the disease, such as obesity, are estimated by the body mass index, synergize with the genetic background, and in particular with this variant in determining hepatic fat accumulation and the risk of mesoly. These are the data from a multi-ethnic population in the US, the Dallas studies. You can see here in the different classes of body mass index, normal weight, overweight, obesity, and severe obesity, there is a large impact of the carriage of the variant in heterozygosity, in orange, and homozygosity, in particular in red, in determining the amount of fat into the liver, hepatic fat content, indicated here, and with an exponential effect, in particular in those with obesity and severe obesity. So this is really the archetype of the impact of genetic variation, and we and others during the last 15 years have demonstrated that the presence of triggering factors for steatotic liver disease, mainly obesity and insulin resistance, but also in the presence of dietary factors such as harmful alcohol intake or excessive intake of fructose, especially in sweetened beverages, or in the presence of even infected triggers of fat accumulation to in hepatocytes, such as the infection with the hepatitis C virus. The carriage of variants was associated with an increased risk accumulating more fat into the liver, developing lipotoxicity, inflammation, and in progression towards cirrhosis, and mainly through cirrhosis, but exerting also a direct carcinogenic activity, in particular in the people homozygous for the variant, to the progression to hepatocellular carcinoma. So consequently, the homozygosity for the methionine variant is highly enriched through the spectrum of liver disease in the population. These are data from the European population, Italian and Spanish individuals will have a similar prevalence of this variant. It's about 5% of the people is homozygous for this variant in healthy general population, but this prevalence increases progressively with the stage of disease. It's 10% in those with steatohepatitis, 16% in those with significant liver fibrosis, and almost 1 in 3 people with cirrhosis or liver cancer due to steatotic liver disease are homozygous for this variant. So it's about an 8-fold absolute enrichment in this genotype, which is striking. I mean, it's almost a unique example in a common condition. So this is really, in effect, comparable to that of a monogenic driver of liver disease, such as the HFE variant of hemochromatosis in the general population, but it's much more common because it's 1 in 3 people is a carrier, and 1 in 20 is homozygous. So these findings have also therapeutic implications. Indeed, especially the group of Ellen Hobbs in Dallas, who first identified the variants and then conducted physiological studies, but also our group in collaboration with Stefano Romeo, then in Dallas and later in Sweden, contributed, demonstrated that the mechanism underlying liver disease encompasses the accumulation of the mutant protein at the surface of lipid droplets in the hepatocytes, because it's involved in the hydrolysis of triglycerides, which is the main component of the core of the lipid droplets and the neutral lipids in the hepatocytes. By the sequestering this essential cofactor, it inhibits the remodeling of the lipid droplets, favoring the accumulation of fat within these lipid droplets, and then reducing the turnover, probably favoring the toxicity. So we hypothesize that by silencing the mutant the variant in carriers, in particular those in homozygosity, we may be able to restore normal lipid metabolism, or releasing the essential cofactor, curing liver disease in homozygous people with progressive liver disease, or preventing the progression in those at the highest risk. And the milestone supporting these hypotheses came with the first studies that clinical study, phase 1-2 studies that use RNA interference to silence PNPLA3 in homozygous carriers of the methionine variants, showing a dose-dependent effect, in this case for an siRNA silencing PNPLA3 in people homozygous for the variant. So after a single dose, you can see that silencing of PNPL3 by this approach was able to induce a dose-dependent decrease in the hepatic fat content, and similar data were obtained by the use of an antisense oligonucleotide. So this possibly represented the prelude of a first genotype-directed therapy in hepatology for a common condition, so a specific subset of people with MASLD at high risk of progression towards liver-related events. Recent work, in particular those with obesity, insulin resistance, which is the main driver of liver disease. So recent work from my group has also revealed another important trigger of the phenotypic expression of this genetic factor in the population, in that we demonstrated that there is a striking interaction between the female sex and the carriage of this PNPL3 variant in determining the risk of liver disease. Indeed, we show initially in a clinical cohort, this is a large clinical cohort, more than almost 1,900 patients with histological mesol D, but we replicated the data again in the population-based UK biobank, that the effect size, so the impact of the variant on the risk of developing the whole spectrum of liver disease, from fat accumulation, steatosis, to hepatocellular ballooning, inflammation, steatohepatitis, and fibrosis, is always larger in women than in men. You can see the effect, the impact of the variant of the risk of steatosis are much higher in women than in men. And this was not an additive effect, of course, also because the female sex is a protective factor against the developing of steatotic liver disease before, especially before menopause, with a very high p-value. And we went on demonstrating that this is due to the fact that PNPF3 is regulated by estrogens through the estrogen receptor alpha. There is a specific estrogen receptor element sequence in the PNPF3 promoter that responds to increasing level of estrogen, inducing the transcription of PNPF3 and then the accumulation of the mutant variant on intracellular lipid droplets, exacerbating the phenotype of the disease, the phenotypic expression of the mutation in women after menopause, where they still have a higher level of estrogens than men, but when insulin resistance can contribute to the induction of the expression of the PNPF3 in the liver and in the development of a peripheral insulin resistance, leading to the increased flux of free fatty acids to the liver. Indeed, when we genotype women with severe mesol D in the population, the homozygosity for the methionine variant is notably enriched. This is a collaborative Italian and European cohorts. You can see that in particular in women with, in the absence of overweight, almost 60% are homozygous in this clinical cohort for the PNPF3 variant, as compared to 8% of the general population. So again, there is a tremendous enrichment in this genetic predisposition factor, especially in women with a lower degree of triggering factor and progressive liver disease, telling us that the steatotic liver disease in women has often a larger genetic contribution. But this not merely shape liver histology, it also predict clinical outcomes. Again, here you can see that especially in women, the homozygosity for the methionine variant, again in the European and Italian cohort, was associated with a higher risk of liver-related events, and this is being confirmed by meta-analysis of the literature. So we have evidence that, in particular, women homozygous for this methionine variant PNPF3 will represent a group that may benefit most from PNPF3 targeted therapies aiming at preventing the progression of liver disease. However, the results of the clinical studies exploring the silencing of PNPLA3 in homozygous individuals were a little bit disappointing concerning the improvement not only of fat but of inflammation and fibrosis. So we need to study more in details, I think, the mechanisms that are leading to progressive liver disease in carriers of the mutation, going beyond the accumulation of fat and study the progression from the fat accumulation to steatohepatitis and inflammation. Unfortunately, mouse models are not so much reliable because PNPF3 is different in men and in mice. The protein has large differences, and even when you knock in the mutation, the phenotype is very mild, so they are not really the best model to study the impact of the variant and to study the PNPF3 targeted therapy. So during the last years with my group, we focused on the exploitation of human liver organoids isolated from surgical tissues, according to a protocol developed by Maristel Uch, and which can be differentiated in vitro, can be expanded, can be biobanked, so collecting biobanks of tissue that can represent the heterogeneity of liver disease in the population and the genetic variation that we see in humans that can be differentiated to hepatocytes with expression of markers of differentiation, and they can be exposed to excess fatty acids. In particular, we use a mix of palmitic and linoleic acid to recapitulate the metabolic environment they are exposed to in the clinic, and they accumulate lipid droplets in the cytoplasm, leading to a mechanism of a model of steatotic liver disease to study the impact of human genetic spontaneous genetic variation on lipid droplets accumulation, and not only on quantitative features but also qualitative features of lipid droplets alteration, which are more difficult to study, of course, in clinical samples and in the clinic. So here we show that this is a work from my lab that still unpublished, that in this model we can recapitulate the strong phenotype, for example, of this PNPL3 variant on the risk of accumulation of fat into the hepatocytes, showing that both in baseline condition and after exposure to a mixture of free fatty acids, the PNPL3 variant has a dose-dependent impact of the accumulation of fat into the cytoplasm of the hepatocytes, here shown by NIRAD staining in green. You can see here there was here highly significant dose-dependent effect on the total lipid droplets area, but in particular on the number of lipid droplets per nucleus, so acting more specifically on the number of lipid droplets than on the average size of lipid droplets, at least in this model, which was all increased in organoids from people homozygous for the mutation. This was independent of any of the other confounders that may impact on the risk of steatosis, the age, the sex of the donor, the presence of cirrhosis, and the other genetic background. A very strong and robust impact in vitro of carriage of this variant on the fat accumulation, which was larger than the impact of the exposure to free fatty acids. And by showing this is a major determinant of the heterogeneity in lipid handling in the liver and in humans in the population. And therapeutically, since we are, our goal is to develop precision therapeutic approaches to stop liver disease, particularly in this high-risk group, we show that the silencing of PNPL3 shown here, the protein is shown here by the red staining by immunofluorescence, by a modified small interferon RNAs that we developed in the lab, similar to that was used in the clinic for the J&J trial, reducing not only PNPL3 protein levels in the hepatocytes, but also the accumulation of lipid droplets, again, consistently with the clinical results. And we were able to show that even in organoids homozygous for the PNPL3 variant, but also in the other genotypes, the silencing of PNPL3 was able to almost normalize the hepatic lipid content, both in the organoids treated with fatty acids and in basal conditions. So recapitulating again the results of clinical trials. Interesting, we got similar results using resmetirom, which is the first liver-directed therapy approved in the clinic. You say we have seen before the data. It's thyroid beta receptor specific agonist inducing lipid oxidation and reducing lipid synthesis in the liver. But resmetirom was not able, it was effective, but was not able to completely normalize intracellular fat accumulation, specifically in organoids homozygous for the methionine variant of PNPL3. So even a high-dose resmetirom was not able to completely correct the phenotype. So what we have tried to do is to combine silencing of PNPL3 with the resmetirom treatment in organoids homozygous for the PNPL3 variant, showing that there is probably an additive effect that we are now confirming, both on the total lipid droplets area and the number of lipid droplets per nucleus, using the combined approach, suggesting and maybe in the future this may be one of the approaches that may be able to further improve the response to treatment in the clinic to the available clinical treatments in very high-risk patients. Of course, this is still the beginning because we show an improvement in amount of fat, but we have to deepen the understanding of what is the impact of PNPF3 silencing on the quality of fat and the activation of the pathways leading to liver damage, including by combining these organoids with non-parenchymal cells to mimic the process of the development of inflammation and fibrogenesis is in this in vitro model, which is not yet recapitulated in a pure parenchymal organoids so far. But if I have time, that there is more than PNPF3 for MESL-D predisposition. Of course, this is by far the main genetic determinant at the population levels, but collectively we have already identified more than 30 genetic variants with the latest large GWAS that specifically predispose to fat accumulation and inflammation to the liver. I again summarize the most important one in the slides, and they illustrate the common themes, such as that most of these are involved in the modulation of the activity or the expression of protein that are involved in lipid metabolism within the hepatocytes, in particular at the level of the intracellular lipid droplets, either in PNPL3, ATGL, or PNPLA2, which is the main triglyceride hydrolase. HSD17B13 also is a lipid protein, a lipid droplet protein expressed in hepatocytes involved in the modulation of lipid metabolism here, or the oxidation of lipids. And in particular, the other pathway that is heavily affected by genes regulating the hepatic fat accumulation is related to the export of the lipids from the liver to the circulation, so the extrahepatic tissue, via the secretion, the synthesis, lipidation, and secretion of very low density lipoproteins. So variants such as those in particular TN6SF2, which are associated with an increased risk of MESL-D. This is the second variants accounting for the second largest fraction of steatotic liver disease heritability, is associated with a retention of fat into the hepatocytes and reduced secretion, in particular triglycerides and cholesterol within very low density lipoproteins. And here distinguishing the correlation with causation is essential to develop treatments and strategies to contrast liver disease progression in liver disease, and by the identification of this panel of genetic variants in MESL-D by Mendelian randomization, which is an epidemiological approach exploiting inherited genetic variation to infer causation between an intermediate phenotype and an outcome in epidemiological studies, because the genetic variants are inherited randomly at conception before birth, independently of causal risk factors. So if all genetic variants cause fat accumulation, also cause fibrosis and cancer, and the effect is proportional, the effect on cirrhosis is proportional to the effect on steatosis, basically through some epidemiological approach, you can demonstrate that fat causes fibrosis and cancer. So this was not trivial 20 years ago, because fat accumulation was deemed to be an innocent bystander in the natural history of liver disease. But by this approach, we show that there is actually very close and robust association between the impact of all these genetic factors on hepatic fat content, here measured by the non-invasive gold standard, which is proton density fat fraction by MRI. This is again data from the UK Biobank, and the risk of both cirrhosis and liver cancer in a very large cohort, but this was also true in clinical cohorts, allowing to conclude that there's probably a causal association between the accumulation of fat and the progressive liver disease. So this is actually a driver, the accumulation of fat into intracellular lipid droplets or progressive liver disease, such as the accumulation of fat into the atherosclerotic plaque is a driver of the complication of atherosclerosis, and as statins, reducing fat accumulation to the plaque, change the natural history of cardiovascular disease. We are therefore hoping that therapies that inhibit the accumulation of fat, or reduce the accumulation of fat into the liver, will then be able to inhibit the progression of liver disease to fibrosis, and in particular liver cancer in the future. This was predicted a few years ago, and actually it turns out that the best therapeutic approach that are being approved right now are mainly acting through the metabolism and reduction of hepatic fat accumulation into the liver, both resmetirom as a main mechanism and even semaglutide indirectly by improving insulin resistance. These genetic variants can also be used to improve risk stratification, because by basically summing the number of risk variants carried by each individuals, which can be weighted by their effect size, so the increase they confer in the risk of liver disease, you can develop polygenic risk scores that summarize the inherited predisposition to develop progressive steatosis, and then we have shown before also cirrhosis and hepatocellular carcinoma, allowing to identify thresholds, so so group of people in the population that are very high risk of developing progressive liver disease, even at the very early stages of the disease, even during childhood, because these are inherited at conception, so you can genotype them very early and be able to predict events throughout the lifetime of the patients in the presence of triggering factors, and they can be incorporated in particular with clinical risk scores, in particular those that also evaluate the metabolic triggers and the stage of progression of liver disease to further improve the risk stratification. So what we have shown is that, of course, not of course, but based on the epidemiological evidence, these polygenic risk scores are able to predict the whole spectrum of progression towards steatotic liver disease, advanced fibrosis, liver cancer in large clinical cohorts and in a population-based cohort, independently of the classical drivers of risk factor of disease, age, sex, BMI, and diabetes, because they are specific for liver-related involvement, and suggesting that we may be able, with further improvement, to use them in the future to identify among people with steatotic liver disease phenotype those with high polygenic risk scores of homozygous, for example, for the PNPF3 risk variant, which may warrant, for example, in people with metabolic disorder, strategies for early mesol D detection, the progression to advanced fibrosis with early access, for example, to therapies or enhanced lifestyle management, because these still respond to, for example, weight loss or improve lifestyle quality, early inclusion for those with more advanced disease in hepatocellular carcinoma surveillance programs. So by identifying a subgroup of people with steatotic liver disease that has a higher risk of developing liver-related complications as compared to the cardiovascular complication of the disease, which, as we said before, are the leading cause of health problems in general in people with mesol D. Identify liver-specific, a subgroup of people with high-risk specific or liver-related complications. To further improve this prediction, we then forwarded, this is mainly work by the Stefano Romeo's lab, and we were collaborating center to develop even more specific polygenic risk score predicting the liver-specific involvement of muscle D, of liver-specific involvement in people with metabolic disorder developing so-called partitioned or process-specific polygenic risk scores, meaning a polygenic risk score of a biological process which is more precise than the whole definition of the disease. In particular, you use a simple measure, fasting triglycerides, which are a proxy for VLDL secretion by the liver, to identify genetic risk factors for steatotic liver disease associated with retention of lipids within the liver, as compared to genetic risk factor for increased fat into the liver that were also associated with an increase in circulating lipids. So a lipid retention component of the predisposition towards liver disease versus a predisposition towards enhanced lipogenesis, systemic insulin resistance, and the systemic complications of liver disease. So basically you stratify the risk variance for hepatic fat accumulation according to their effect on the baseline triglycerides. And what we could demonstrate was that indeed the risk of liver disease was particularly enhanced in those with these discordant polygenic risk scores, encompassing the evaluation of genetic variant associated with retention of lipids into the liver, including PNPLA3 and TM6SL2, the major genetic determinants, which increase much more the risk of chronic liver disease, cirrhosis, and liver cancer, but protected against cardiovascular disease because they reduce the secretion of cholesterol and lipids into the circulation, so they shift the balance, tip the balance towards the liver-related versus the cardiovascular complication of the disease, whereas the systemic component of the disease was also associated with a milder increase in liver events, but also with an increased risk of developing the cardiometabolic complication, cardiovascular disease, heart failure, diabetes, hypertension, and even chronic kidney failure. So we can identify right now probably two subtypes, or at least two different components of MESOL-D, even from the genetic point of view. One that is predisposed to systemic insulin resistance and the systemic complication of liver disease and enhanced lipogenesis, and one which is more liver-specific, associated with the retention of the lipids into the liver and leading to the liver-related complications of the metabolic dysfunction. We can probably do even better in the next year by also incorporating the evaluation of rare genetic variants into this genetic evaluation, because right now with the GWAS we could evaluate the role of a common inherited variation. But as the recent meta-analysis of GWAS in the literature shown, there is probably also a role of some low-frequency variants and rare variants in determining, in particular, severe phenotypes of liver disease in some specific families, because these variants are rarer in the population, so they account individually for a smaller fraction of liver disease risk, but singularly they increase, they have a larger impact on the risk of developing liver disease in the single individual of families, because they have a larger impact on on the function of the protein they encode. So we think this is important, for example, especially in families of patients with advanced MESOL-D, because we know that there is strong familial aggregation of casein casein progressing to a severe chronic liver disease in the population. In particular, first-degree family members of people with advanced fibrosis due to MESOL-D have a higher risk of developing steatotic liver disease as compared to the general population, more than 3-fold times higher after correction for age, sex, diabetes, and body mass index, so after correction for the metabolic risk factor. But in particular, they have a much higher risk of developing advanced fibrosis as compared to the general population, a more than 15-fold higher risk of developing advanced liver fibrosis compared to the general population. highlight an important role of a familial inherited factor, both genetic and probably epigenetic or metagenetic due to the microbiota, possibly also in determining progressive liver disease. And this was not was partially dependent of carriage of PNPF3 variant, but was not accounted for by common genetic variation. So what we hypothesize, and this will be my last part of my talk, is that rare genetic variants shared among families may account for part of this, let's say, missing heritability of liver disease, and might help us to explain the aggregation of cases with progressive liver disease due to steatotic liver disease in specific families. This was a first study we published in 2019, where we, by whole exome sequencing, examined rare genetic risk factors influencing hepatic lipid metabolism in people who develop hepatocellular carcinoma due to MESL-D, showing that there was an enrichment in missense and likely damaging variants in people with hepatocellular carcinoma as compared to the general population. In particular, in this gene, not surprisingly maybe, which is ApoB, which is the key protein, the gene that encodes for the key protein involved in the secretion of very low density lipoproteins from the hepatocytes again. And these data were supported by findings, again, these are data from the UK Biobank, from the general population, showing that by conducting a rare variants associated study in the whole genome, the variants again in ApoB and microsomal triglyceride transfer protein again involved in lipidation of very low density lipoproteins by hepatocytes and of course by the duodenocyte as well, were the main determinants of the risk of developing steatotic liver disease or increased hepatic fat content in the general population. So again, suggesting that an impairment of the secretion of lipids by the liver is a main driver of the process that leads to steatotic liver disease and progressive liver disease. Now we'll finish with this data because the paper has been accepted yesterday, so I can show you the results. Because we now collected a larger cohort of people, more than 500 people with significant liver fibrosis due to MESL-D with available whole exome sequencing data. And we could confirm in this cohort of Italian and European patients that there is a stronger enrichment in loss of function and damaging variants in ApoB as compared to the general population, which a more than 10-fold increase in the risk, almost 15-fold, depending on how you define pathogenic mutation as compared to the general population, and also identify a specific threshold of LDL cholesterol that is associated with the carriage of such mutation, because the carriers have a phenotype which is consistent with heterozygous familial hypo-beta lipoproteinemia. So not only this is a risk factor for progressive liver disease, but you have a simple phenotype that is routinely measured in all patients that can allow you to identify those that are at higher risk of being carriers of such a genetic determinants of liver disease. And in our cohort, 1 in 20 patients with advanced liver fibrosis due to MESL-D was a carrier of this genetic mutation. So importantly, by conducting the family study in this clinical cohort, in the relatives of patients with advanced liver disease in the clinical cohort, and the meta-analysis in the whole literature that is available for heterozygous familial hypolipo beta proteinemia and the risk of liver disease, we could also demonstrate that there was an overtransmission of the risk variants in family members affected by steatotic liver disease, severe steatosis, of liver fibrosis, and a borderline significance for clinically significant liver fibrosis, also demonstrating that this is probably one of the key factors that may account for the familial aggregation of cases with progressive liver disease in the population in clinical cohorts. So once you identify a potential carrier in your clinical cohort, it may be also worth to study the family members, in particular first-degree relatives, because you can identify carriers at high risk of developing liver disease in the future in the presence of triggering factors. And by considering these clinical cohorts and meta-analyzing the data with the population-based UK Biobank and with the Million Veteran Program cohort in the US, both the European ancestry participants and African American participants, we could show that and confirm that this is a main the risk factor for the risk of developing cirrhosis in the population, robustly increasing the risk by about 80%. 1% of the general population is a carrier of damaging variant in ApoB. This increases the risk of cirrhosis by 80%, and increasing the risk of developing hepatocellular carcinoma by more than 3.5-fold. So this is a relatively rare risk factor for liver disease, but is a large effect on the risk of progressive liver disease. We also show that there is and this may be interesting for those that are studying the role of the intestine in the pathogenesis and the gut-liver axis in the pathogenesis of progressive liver disease, because we show for the first time that there is different impact of mutation that affect specifically ApoB100, so the section of the protein that is expressed only in the liver, the second half of the protein, as compared to variants affecting the first part, ApoB4, which is common between ApoB48, which is expressed by duodenocytes and involving the secretion of chylomicrons, and ApoB100 in the liver. So basically, mutations affecting both ApoB48 and ApoB100 have a larger impact on the risk of liver cancer, despite the metabolic phenotypes, both the impact of circulating triglycerides, the risk of coronary heart disease, the accumulation of fat into the liver, and the other metabolic phenotypes were more severe, not more severe, but had a larger phenotype in the carriers of the mutation specific for the ApoB100. In that case, it's protective because the mutation in ApoB100 associated with a strong reduction in triglycerides, LDL cholesterol, increased HDL cholesterol, and an increase in hepatic fat content, but they protect against coronary artery disease. So basically, from the clinical point of view, we can go and right now with the clinical availability of next generation sequencing, which is becoming more and more available at least in the referral centers in Milan, but I guess also in London and here in Madrid and the main clinical centers, we can detect the presence of variants in ApoB and other genes involved in lipid metabolism in patients meeting some specific criteria with steatotic liver disease and specific phenotype. The first one I told you before had a strong family history in the first family member, but also the age of the early age at onset, and the absence of a triggering factor for the disease are red flags that may want you to look for the presence of strong genetic determinants of steatotic liver disease. Can also interact with obesity and the other environmental factor for steatotic liver disease. Using this approach, what we found in our cohort in Milan is that almost 1 in 3 people meeting those criteria actually as a carrier of a genetic variant or a risk genotype, allowing you to diagnose a genetic condition which is actionable and may change the clinical management of the condition. The most common ones are indeed heterozygous familial hypo-beta lipoproteinemia, carriage of mutation of ApoB, but also other causes such as phenotypes predisposing to hepatic iron accumulation, some telomere-associated disease, hereditary fructose intolerance, or other, or cholesterol ester storage disease are other common causes that can be detected by this approach. So by conclusion would be that genetics is no longer an academic and basic research exercise, but is becoming an integral part of risk prediction through the use of polygenic risk scores, but also through the advent of next generation sequencing in the clinic, also of counseling, family screening, and also for the identification of drug potential therapeutic targets. So it's an instrument that is available for the pathologist, that it enriches and will not replace, of course, the clinical evaluation based on biochemistry, biomarkers, elastography, the imaging, liver histology, but it's something that can add to help us to understand better the complexity and the heterogeneity of the disease and give us additional tools to prevent in the future the complication of liver disease. With this, thank you. It was my last slide. Thank you for your attention. Of course, I should thank the members of my group involved in the molecular biology and cellular lab, the omics and bioinformatics lab, and involved also in biobanking, processing of samples, and so on. My long-standing collaboration, especially Stefano Romeo, with whom I share most of the studies on PNPF3 and the genetic liver disease during the last 15 years. And you for your attention. This is for those that were yesterday, our hospital in downtown Milan. This is here, Piazza Duomo, and this is the ancient hospital of Milan, which is now the site of the university, founded in 1456, first public hospital in Europe, and this is the new hospital that will be open this year. I hope to be able to host you in the future, and thank you. So of course, you want to target the protein where this has a detrimental effect, so in all cell types where it's involved in determining pathological phenotype. So we know that broadly the impact correlates with the accumulation of fat into the liver, but there are convincing data from different groups, independent groups, including our group, that the variance is also a phenotype, for example, in hepatic stellate cells, because it's probably more expressed in hepatic stellate cells than in hepatocytes. And the hepatic stellate cells is involved in the probably the hydrolysis of retinol esters from intracellular lipid droplets, including after the activation of the hepatic stellate cells, that you know that they lose the lipid droplets and transactivate and proliferate and so on following liver damage. So, and of course, retinol, once it's released, has a broad effect in the regulation of gene transcription, regeneration, and inflammation into the liver. So basically, the hepatic stellate cells, primary human hepatic stellate cells carrying the variants, particularly in homozygosity, they have a more pro-inflammatory, pro-fibrogenic phenotype. This has been robustly demonstrated. But to what extent this account for the risk of progressive liver disease, I don't know if 50% of the effect is due to hepatic stellate cells or just 5%, we don't yet know. But it's likely to be involved. Concerning sinusoidal endothelial cells, there are less data, but I remember, and still curious about it, first GWAS, so genome-wide association study, was circulating ICAM1 levels, and this PNPF3 variant was the main genetic determinant of circulating ICAM1 levels in the population. So Yeah, actually, we first reported the association with the risk of hepatocellular carcinoma independently of cirrhosis in patients with chronic hepatitis C back when it was the most common cause. It still is a major cause of hepatocellular carcinoma, even after eradication, and at least in Southern Europe, but definitely is a serious factor for progression to hepatocellular carcinoma, also in chronic hepatitis C. It has a larger impact in pure metabolic disease and in alcoholics liver disease, and of course in the mixed forms, because they are less viral cofactors, it's more purely metabolic cofactors. So it's more enriched if you select patients with hepatocellular carcinoma due to MASH or ALD, you will find a larger enrichment, especially in homozygosity for this variant than in those with chronic hepatitis C, and in particular those with chronic hepatitis B, where it's not probably playing a major role. Because for chronic hepatitis C, there's probably also synergy in inducing fat accumulation, which is probably one of the mechanisms leading to progression of also chronic hepatitis C patients. Concerning the impact on the prognosis, there are not good quality data, because we also published some years ago some data in our retrospective cohort, even before systemic treatments, but there are no data in a well-characterized cohort with homogeneous stages of the disease and homogeneous treatments. So, if you take a cohort and you know very well that the outcome is heavily affected by the stage at which the HCC is diagnosed and the treatment and so on. So we don't have the high-quality data to be able to say whether or not it affects the prognosis of the patients right now. Yeah. PNPL3 animal, the knockout has no phenotype suggesting, but we know that the PNPL3 has a different role in regulating lipid metabolism in the mice as compared to man, because when you knock down PNPLA3 in mice, you have an alteration in the quality of lipid droplets that is similar to the one that you see in carriers of the variants in humans, but these are not phenotype on the hepatic fat content and liver disease in mice. Whereas in mice, you have a phenotype when you knock in the human mutation and you have the mild accumulation of fat, but not inflammation and fibrosis. So it's not really... So, it's useful to study some of the mechanisms related to the accumulation of fat, but not to study the whole story, I think. And also retinal metabolism is very different between mice and humans, so probably also the hepatic stellate cells component of the disease predisposition cannot be really recapitulated in mice. So Eleanor's group in Dallas is using mainly mice to study the pathophysiology, and they propose and convincingly demonstrated that in mice the accumulation of fat in the hepatous is dependent on the accumulation of the mutant protein on the lipid droplets, which is hindering the remodeling of the lipid droplets. And this is convincing in mice, but I think in humans there is more to be. It's already starting with bird cohorts in some regions of Italy, in the UK, the large population-based cohorts. So we will know our risk genotype, and we will be able to determine this polygenic risk score very early during the lifetime. So I think the earlier the better, because you can you are more likely to be successful with lifestyle changes, for example, in children with obesity than in 65 years old women with diabetes, hypothyroidism, and arthritis. We cannot exercise anymore. And of course, the earlier the prevention is to prevent is better than to cure the disease. But right now, I think may be useful in those. We have seen that we begin to see some effect in the medium term after 3 years in those with an altered FIB4, those with metabolic risk factor and a FIB4 higher than 1.3. And then after 4 years, you begin to see divergence of the cure of the incidence of liver-related events. So because most of the effect of the genetic variants can be mediated by fibrosis, so you can see it with FIF4, but it also predicts the progression during the follow-up. So after a few years, you will see a higher risk in those with high FIF4 and the variant as compared to high FIF4 without the variants. And the difference is larger if the FIF4 is already above 2.5, for example, because the risk of events will increase at earlier time points. Yes, it is possible. That is good news, at least for the PNPL3 variant. They may actually benefit more from lifestyle intervention because the baseline risk is higher. So if you follow, adhere to, for example, Mediterranean dietary pattern, you have a larger benefit in terms of the risk of liver events if you are a carrier of the PNPL3 variant, or if you have a high polygenic risk score. Yes, so there is a benefit. That's good news. So in the absence of triggering factors, the homozygosity for the PNPL3 variant is not associated with liver disease. So you are perfectly healthy if you don't drink, if you are not overweight, you are not exposed to other hepatotoxic factor, you are perfectly healthy. So meaning that if you are overweight and a mild steatosis, but you can reverse the overweight and improve your diet and increase the exercise, you can reverse this risk. Yeah, at the early phases, of course, once cirrhosis is already established, it's a different. I don't think we have enough data to answer definitely this question. I remember that at the beginning we studied in collaboration at the time with Valerio Nobili in Rome, pediatric patients with MASH, and there was an even larger role of this genetic predisposition as compared to adults. So I think it may be useful at that stage to identify those at highest risk of liver-related events. But of course, I don't think that at this stage it will change therapy, because in any case you will try to make them lose fat and improve their metabolic health, and you won't treat them with liver-directed drugs during childhood. Maybe if when we will have evidence that these people can benefit from early establishment of liver-directed therapies, young adults may be a group that may benefit most from this approach. Yes, I think this is the next step, probably, because if the silencing, so what's the problem of silencing? Maybe that in humans, the enzymatic activity of the wild type is also important to remodel the lipid droplets in the hepatocytes. So there is not only a role of the accumulation of the mutant protein, but also the loss of the enzymatic activity of the wild type. So this won't be restored by the silencing of the mutants. So the approaches that are being developed right now are mostly related to RNA editing, trying to correct the translation of the protein, the amino acid at the level post-translationally with RNA editing approaches. But of course, the genomic editing will be another possible option in those with severe disease, of course, because I mean, right now probably it's too much. We are not yet ready to use genomic editing for I mean, it's not a monogenic disease with where the phenotype is only determined by the single strong genetic determinants, but for complex diseases, I don't think the regulatory agencies are ready to accept a gene therapy for a complex disease. I think it may benefit still a bit, but not resolve all the problems. So the RNA editing is right now the possible approach, the most likely approach. Yeah.
Thank you very much for your talk. Thank you for your questions. And here we conclude this liver seminar winter edition, and we will see you in the summer edition in Barcelona. Thank you.